All right, let's just jump right into this explainer. Today, we're demystifying one of the most elegant, high-speed, and frankly mind-boggling processes in all of biology, DNA replication. If you've ever found yourself wondering how on earth a cell manages to perfectly copy millions or even billions of letters of genetic code right before it divides, well, you are definitely in the right place. We're going to pop the hood on this microscopic machinery and see exactly how it gets the job done. To make sense of this whole intricate biological dance, we're breaking it down into a logical roadmap. First up, the elegant copying hypothesis. Then we'll look at proving the semi-conservative model. After that, we dive into the molecular replication factory itself. And finally, we'll finish up by navigating the replication fork.
So let's kick things off with Section 1, the elegant copying hypothesis.
Take your mind back to 1953. James Watson and Francis Crick have just published their now famous model of the DNA double helix. And at the very end of their paper, they dropped this understated yet incredibly iconic quote. Basically an absolute mic drop moment for biology. By realizing that adenine always pairs with thymine and guanine always pairs with cytosine, they completely set the stage for how life passes on its genetic instructions. The physical structure of DNA literally contained the secret to how it could be copied. They called this proposed scheme semi-conservative replication. Picture the original double helix unzipping. As those two strands separate, the strict, unbreakable rules of base pairing mean that each individual strand acts as a completely flawless template for building a new partner. The real crucial takeaway here is that half of the original DNA molecule is quite literally conserved in every new copy. When a cell divides, it doesn't just build a brand new double helix from scratch. Instead, the two resulting DNA molecules each have one old parental strand and one completely new strand. Honestly, it is just a remarkably efficient way to make sure the genetic code stays intact across generations. Moving on to Section 2, proving the semi-conservative model. Because, a beautiful theory is great, but in science, it's not enough. How did they actually prove DNA replicates like this? Well, the answer came a few years later in 1958. Matthew Meselson and Franklin Stahl designed this brilliantly clever three-step experiment. They needed a way to tell the old DNA strands apart from the new ones, right? So they used isotopes of nitrogen. Step 1, they grew E. coli bacteria in a medium with a heavy isotope of nitrogen, 15N. That made the bacteria's DNA physically heavier. Then, step 2, they moved those cells into a normal, lighter 14N medium and just let them replicate. Finally, step 3, they extracted the DNA and spun it in a centrifuge to separate it strictly by density. And the results were stunning. After exactly one generation in that lighter nitrogen, the DNA they extracted wasn't entirely heavy and it wasn't completely light either. It formed a distinct band right smack in the middle. It was a perfect hybrid. This meant every single DNA molecule had one heavy parental strand and one light, newly synthesized strand. Boom. Unequivocal proof of the semi-conservative model. Just to really solidify this, it's not just a bacteria thing. That same year, Taylor and his colleagues ran similar experiments using radioactive thymidine to track newly built DNA in fava beans. They proved that the much more complex chromosomes in higher organisms, like plants and by extension us humans, replicate in the exact same semi-conservative way. The rule holds true across all of biology. All right, Section 3, the molecular replication factory. Now that we know the theory is solid, let's zoom way in on the high-tech, slightly chaotic reality of what's happening inside your cells right at this very moment. I want to introduce you to the primary worker on this factory floor, DNA polymerase. Specifically, DNA-dependent DNA polymerase. This molecular machine has a ridiculously demanding, high-stakes job. It reads the existing DNA template strand and chemically links together the new individual building blocks, the deoxynucleotides, to form the brand new complementary strand. To really wrap your head around the scale of this enzyme's job, just look at this number, 2,000. That is the staggering number of base pairs an E. coli DNA polymerase processes every single second. Imagine trying to copy a book by typing 2,000 letters perfectly every second. That's the kind of speed and precision we're dealing with here. To put that into perspective, an E. coli bacterium has a genome of about 4.6 million base pairs. Thanks to the blinding speed of DNA polymerase, the entire replication process for the whole genome is done in literally 18 minutes. I mean, in the time it takes you to brew and drink a cup of coffee,
This microscopic machinery pulls off an absolute masterclass in biological engineering. But obviously, building millions of base pairs that fast, it requires a massive amount of energy. So how does the cell handle it? Well, it solves the problem brilliantly. The building blocks themselves, the deoxyribonucleoside triphosphates, serve a dual purpose. Not only do they act as the physical letters added to the new DNA chain, but they actually bring their own battery pack. Their terminal high-energy phosphates break off during the process, and that releases the exact burst of energy needed to power the reaction. It's a completely self-powering, incredibly efficient system. Finally, let's hit section 4, navigating the replication fork. We need to look at how this machinery handles the physical hurdles of actually unzipping and copying a tightly wound double helix.
Because those two long strands of DNA cannot just be completely pulled apart all at once. Replication happens inside a tiny, localized opening. It's literally a fork in the road, where the double helix splits open just enough for our machinery to get in and go to work.
Now, the factory doesn't just start randomly unzipping the DNA wherever it feels like it. It has to begin at a highly specific starting line known as the origin of replication. Think of this as a defined landing pad in the DNA sequence where the initial enzymes can attach and create that very first replication fork. Without it, organized replication simply wouldn't happen.
But here is where we hit a really fascinating biological quirk. DNA polymerase is an absolute strict rule follower. It can only build a new strand in one specific direction. But, as we know, the two strands of a DNA helix run anti-parallel. They face opposite directions. So, on the template strand running 3' to 5', replication is continuous. The polymerase just smoothly cruises along following the unzipping fork. But on that opposite strand, the one running 5' to 3', the polymerase has to constantly jump backward and build in fragmented chunks as the fork opens up. This is what we call discontinuous replication.
Of course, leaving a cell with a chopped-up, fragmented DNA strand would be completely disastrous. So, to clean up that discontinuous strand, a new vital worker steps onto the floor: DNA ligase. You can think of DNA ligase as the molecular glue. It cruises along that discontinuous strand and seamlessly bonds all those loose fragments together, making sure the final product is one continuous, perfect piece of double-stranded DNA. So we've seen how elegant, how fast, and how incredibly precise this biological factory really is. But that actually leaves us with a pretty critical thought to end our explainer on today. Considering the blinding speed we talked about, thousands of base pairs every single second, and the sheer physical complexity of all this unzipping and copying, no system is 100% infallible, right? So what does it actually mean for an organism, or even an entire species, when this magnificent machinery inevitably makes a mistake? It's a question that drives the very core of evolution, not to mention genetic disease. Definitely something to chew on the next time you think about the unbelievable machinery of life ticking away inside you. Thanks for joining me on this deep dive.